Hey guys, so this is the video about meiosis that I told you guys I was gonna make for you. We talked about meiosis in class, and we described all the phases of meiosis. We talked about the purpose of meiosis, and we compared it to mitosis as well. But we'll go step by step, and I'll try to explain it again. That way, you can watch the video a few times if you need to, if you're confused, or you can come to tutoring. Okay. Remember, tutoring's you're make sure you know the tutoring times, and just come to tutoring, and we can always go into more detail or try to explain it a different way. Okay, so meiosis, like we said in class, is divided into two divisions. Remember, meiosis has two divisions. The cell divides into two, or two times. Mitosis divides only one. But meiosis is divided into meiosis 1 and meiosis 2. Right here, this one's describing meiosis 1. And what happens here, the general idea is that the homologous chromosomes, or same chromosomes, remember you get one from mom, one from dad, okay, they separate and they make two genetically different daughter cells. And the reason they're different is because of this process right here that happens during prophase number 1. Okay, so prophase number 1, this is where crossing over happens, and this is the crossing over right here. You can see it very well in this chromosomes right here. You see that the blue one actually has a small part of the green one. Okay, so they exchange information. That's crossing over, and that's why siblings, even though they look similar to each other, your brother or your sister, they kind of look the same, but they're not identical unless they're identical twins. Okay, and that happens because of crossing over. So this is happening to germ cells that are going to turn into gametes or sperm and egg cells. Okay, so that's meiosis 1. So in meiosis 1, again, the chromosomes separate and two identical daughter cells form. That's the end result of the process. Now, meiosis 1 is divided into 4 phases, just like mitosis. Remember that. Okay, it's just that meiosis 1, all the phases in meiosis 1, they all going to have a number 1 or a Roman numeral number 1, which tells me or tells whoever's looking at this that this is happening in meiosis 1. Okay, so this is prophase 1, this is metaphase 1, this is anaphase 1, and this is telophase 1. Okay, and we'll go over each phase in a little bit, but and again, we're just going to go through the descriptions of them. If you're still confused, make sure you come to tutoring so we can talk more about it. So prophase 1, which is the first thing that happens in meiosis 1, again, the chromosomes pair up, and you can see it here. Okay, so these chromosomes are going to start pairing up, and the nuclear envelope, okay, which you can see here, starts breaking apart, right? It starts dissolving, just like in mitosis. But the big difference between mitosis and the prophase in mitosis and prophase 1 is that crossing over occurs or happens, and that's what makes the cells different. Okay, so that's the big idea behind prophase 1. Crossing over happens, the nuclear envelope dissolves, these chromosomes start pairing up. Okay, now the next step. Okay, or before we even go to the next step, we talked a little bit about crossing over, and what that is the exchange of information between the chromosomes, the homologous chromosomes. Okay, and again, that results in genetic diversity, meaning you're going to have different genetics. Every cell is going to have a different set of instructions, and that's a good thing so that we can have different genes. Everybody can be different and increase the diversity so that we can also increase our survival rate in the planet, okay, or in the environment. Now, the next thing that we also talked about was independent assortment, saying that chromosomes, when they line up during metaphase, okay, they line up randomly. Okay, sometimes they're going to line up like this picture right here. Okay, the blue ones on this side, the red ones on this side, and then they're going to split, and that's how the gametes are going to look. But sometimes they're just going to you're going to end up with a red and blue. Sometimes you're going to end with a blue and red, depending on, just on how the chromosomes line up. It's random. It's not, it's not always the same. It's always different. Again, that makes us unique. That gives us our own genetic instructions. Okay, and that happens during metaphase 1. That's where the chromosomes line up in the middle. Okay, and independent assortment happens. So you can see it here, just like in metaphase, these chromosomes are lined up in the middle, and they're getting ready to go to opposite sides. Okay, and that's what happens in anaphase. And in anaphase 1, homologous chromosomes, okay, not chromatids, but the entire chromosomes. You can see here, one chromosome's going this way, the other one's going this way. Okay, the chromosomes separate and they move to opposite sides. Okay, and after they go to opposite sides, then we're going to have our next phase, which is telophase or cytokinesis. Telophase cytokinesis, that's going to develop two, you're going to end up with two cells, okay, that are going to be different. They're going to be, at the end of these cells, you're going to have, or these cells are going to have 46 chromosomes still, okay, and they're going to be diploid cells. So, and what that means is that they have two sets of chromosomes, just like you should have in your notes. Diploids, two sets of chromosomes, 46 or 23 pairs, okay? So after this, the first division happens, or telophase and cytokinesis 1 happens, then we go into the next division, which is meiosis 2. And this is where the chromatids separate and four genetically different cells are formed, and this is where we get our haploid cells, and you can see them here. Again, this is the big picture. This is the entire process of meiosis 2 in one slide, okay? All right, so let's go through prophase 2. In here, the nuclear envelope dissolves again, and the chromosomes, the spindle fibers are formed again, and the cell is getting ready, or the chromosomes start getting ready to line up in the middle. Okay, there's no crossing over in prophase 2, only in prophase 1. After that happens, after the nuclear envelope dissolves, then you go to metaphase 2. And this is where the chromosomes line up along the equator. And if you see, there's only a set of chromosomes. There's not a pair like in metaphase 2. Okay, so in metaphase 1, it's just single chromosomes line up in the middle, so that during anaphase, the chromatids, okay, you can see it here, only half of the chromosome goes to opposite sides. It's not, if you remember in anaphase 1, okay, there's chromosomes going to opposite sides of the cell, okay, or an entire X, if you want to think of it that way. In here, or in anaphase 2, is only half of the X. So half of the X is going to that side of the cell. So anaphase 1, anaphase 2. Anaphase 1, chromosomes move. Anaphase 2, chromatids separate. Okay. After that happens, then telophase and cytokinesis 2 happens, and that's when you end up with your four haploid cells, which are genetically different. Okay. There is no none of the cells are going to be identical because of crossing over, and that's what gives us our genetic diversity. Okay, so that is something that's what makes meiosis unique, and that's the purpose of meiosis. The purpose of meiosis is just to make sex cells or gametes that are genetically different, so that we can have genetic diversity in any population that we're talking about. Okay, so again, diversity is very important to any organism and for its survival. Now, we also looked at this drawing and we simplify the number of chromosomes at each step, and we compare mitosis and meiosis. Now, mitosis starts with a cell or a body cell, somatic cell, that has 46 chromosomes. Then it divides one, and it goes through this the four phases that we mentioned earlier, or the acronym PMAT: prophase, metaphase, anaphase, telophase. And at the end of mitosis, you're going to have two cells that are identical to the original one. Okay, both are going to have 46 chromosomes. And they're also going to be diploid. Okay, so there's one division in mitosis. It results in two identical cells that are diploid, meaning that they have two sets of chromosomes. So that gives you a total number of 46 chromosomes, which are 23 pairs. Okay, now the process that we just learned, or that we talked about, was meiosis. And meiosis, just like mitosis, starts with cells that have 46 chromosomes. These are called germ cells. Now these cells are going to divide once through meiosis 1, okay, and it goes through the same thing, PMAT 1, prophase, metaphase, anaphase, telophase 1. And at the end of meiosis 1, you're going to have two cells that have 46 chromosomes. But the difference between meiosis 1 and mitosis is that meiosis 1, crossing over happens. So these cells are going to be different than to the original cell. Okay, so when meiosis 1 happens, then meiosis 2 will happen, and those two cells will divide, forming two cells each, with an end result of four gametes or sex cells that have 23 chromosomes each. So that when you get one from your mom, one from your dad, or in any organism, uh, when they fertilize, you'll get half from the male, half from the female. Okay, and this is called meiosis two. And the same phases, the cell goes through the same phases: prophase two, metaphase two, anaphase two, telophase two, cytokinesis. Okay, now meiosis, as a in general, meiosis, both divisions, okay, has two divisions: meiosis one, meiosis two. We said that it creates four genetically different cells, which are haploid, meaning half of the original information, half of the genetic information that was in the original cell. Okay, so that's meiosis one and meiosis two. Now remember and don't forget that crossing over happens in prophase one, not in prophase two, only in prophase one. And what that is this example, you have the two chromosomes, so the red and the blue chromosome, and what they're going to do is just going to exchange a little bit of information, and now they're going to be unique. They're going to be different, okay? And that's what happens during crossing over. Another thing that you need to remember is that chromosomes separate during anaphase 1 of meiosis, and chromatids separate during anaphase 2 of meiosis. During mitosis, you don't need any number after the phase. Chromatids separate during anaphase. Okay, so again, make sure you go back and watch the video, look at your notes, and get ready for your quiz when you come to class. Okay, I hope this video helped. If you still need some help, make sure you come by tutoring.